My name is Julius Lucks. I'm a professor at Northwestern University in the Department of Chemical and Biological Engineering and a member of the Center for Synthetic Biology. So my lab and I are really interested in RNA molecules. So you may have heard about DNA, kind of the double-stranded polymer that's nature's hard drive, but you may not have heard about its close cousin RNA molecules. Now, RNA is really interesting because they're a lot like DNAs, except they're single-stranded. And they look like spaghetti molecules in the cells that kind of wiggle and jiggle and fold into all sorts of different knots and shapes, kind of like a kitten playing with a ball of yarn. What we find interesting about those structures is that each different shape of the RNA molecule does something different for the cell. And what scientists are trying to figure out is how all those different shapes give rise to the many different things that we observe in life around us. So to get at that, my team and I developed a technology that has a fancy name called ShapeSeq. And essentially what it uses is the same technology that people are sequencing their DNA genomes with, but instead of getting the genome sequence, we try to get the shape of the RNA molecules. And the questions that we're really fascinated about is not only what these shapes are, but how they change in time, how these shapes kind of morph into different structures and allow the cells to perform all sorts of different things, from controlling metabolism and what we eat, to moving different places, and how these shapes might even contribute to new cures for certain types of diseases.